Estrogen's metabolism through oral intake. Estrogens, otherwise known as oestrogens, are a family of C18 steroids that are responsible for the development and maintenance of female characteristics and reproductive cycles. Estrogen exists naturally as a steroid hormone as well as synthetically. Naturally, estrogen is produced by the ovaries and is responsible for the function and maintenance of female organs such as the uterus, ovaries, and breasts. Estrogen is not exclusively found in women. The hormone is also produced in small quantities in men. Synthetically, estrogen is used as a hormone replacement drug, particularly pre- and post-menopause, as well as in combined oral contraceptives. As humans, we come in contact with estrogen through oral intake in our daily diets, particularly through fruits and vegetables, as well as in dairy and grain products. The highest dose of estrogen that is taken orally is through medications such as birth control and other hormone replacement drugs. For this particular study, we will be looking at the metabolic pathway of the synthetic form of estrogen, 17-beta-ethinylestradiol, otherwise known as EE, starting from the oral intake. Ethinylestradiol is the most common component ingested through contraceptive pills, and the absorption properties are quite similar to the naturally existing estrogens. As with the absorption of the natural estradiol, the toxin is largely hydrophobic due to the ring structures. It also has two polar hydroxyl groups and an ethinyl chain. Ethinylestradiol, like the natural hormone, has a high log POW value in the range of 3.63 to 3.9, indicating it is hydrophobic and largely bioavailable, as well as a high pK value of 10.33 at the strongest acid. It will therefore remain protonated and leave the molecule neutral, resulting in transcellular diffusion, making it not transport dependent. Ethinylestradiol is completely absorbed rapidly and has a bioavailability of approximately 40%. After ingesting a contraceptive pill or a hormone replacement tablet, the distribution of the exogenous ethinylestradiol throughout the body occurs rapidly. The toxin passes through the epithelium of the gastrointestinal tract and is absorbed in the small intestines. The ethinylestradiol then passes through the hepatic portal vein and enters the liver where it reaches its target, the hepatocytes. The toxin then undergoes pre-systemic metabolism during the hepatic first pass, lowering the bioavailability of the drug further. From the liver, the metabolized estradiol circulates throughout the blood, bound to proteins such as the plasma protein albumin, as it passes through various organ cell membranes to reach the estrogen receptors. Estrogens are distributed throughout the body in the secreted bile acids released by the liver through enterohepatic circulation, resulting in an additional reabsorption of the toxin. Naturally occurring estrogens are distributed throughout the body, however they are heavily concentrated in the sexual organs and tissues. The distribution of the ethinylestradiol is important because there are receptors in numerous organs throughout the body that require interaction with the hormone. If the drug is restricted to one organ, there would be consequences. Estradiols, both synthetic from an exogenous standpoint and natural from an endogenous standpoint, are metabolized in similar fashions. The metabolism of ethinylestradiol takes place in the liver and is promoted by isoforms of the cytochrome P450 enzyme. In phase 1 of the metabolism, oxidation reactions at the 2, 4, 6, and 16 beta carbon locations are predominantly catalyzed by cytochrome P450 3A4, however P450 2C9 can also be a contributor. Both isoforms are found in the hepatocytes. Oxidation at the carbon 2 location is the most predominant. The metabolite 2-hydroxyl-ethinylestradiol is quantitatively the most dominant as it makes up close to 90% of the metabolites formed. In natural estradiol, the 16-alpha-hydroxyl is the most dominant, however due to the steric hindrance of the ethanol group at C17, in ethinyl estradiol, the beta formation of the 16-hydroxylation is not often observed. To the right is the molecular structure of ethinyl estradiol, with the carbon locations numbered for easier visualization. Phase 2 is crucial for the hydrophobic toxin. Ethinyl estradiol can undergo a number of different conjugation reactions due to the nucleophilic hydroxyl groups at either the 3 or 17 carbon positions. Glucuronidation makes for the fastest excretion of the toxin, as it quickly makes it inactive and undergo renal clearance. Glucuronidation has the electrophilic leaving group of UDP that the nucleophilic oxygen of the hydroxyl group will attack. Sulfonate conjugation occurs through the sulfotransferase catalyst, and it too is attacked by the nucleophilic oxygen in order to kick out the leaving group. The sulfonate conjugates are not as easily excreted as the glucuronides. The sulfonates recirculate through enterohepatic recirculation at a concentration of 10-fold higher than the unmetabolized ethinylestradiol and are reabsorbed. Methylation can also occur as it caps the hydroxyl group at the 2-carbon position, preventing the metabolite from being excreted at all. The estrogen metabolite then goes to bind to estrogen receptors and will further transcribe genes. Methylation acts as an inhibitor to toxic effects as it blocks the ability for the metabolite to undergo further oxidation that could result in possible toxic outcomes. On the left is a diagram of natural estradiol, which is being metabolized to 2-hydroxylestradiol and then conjugated to 2-methoxyestradiol. Metabolism affects the distribution as a result of a conjugate pathway. As previously explained, metabolites that are conjugated with sulfate conjugation are recirculated through enterohepatic recirculation at a higher concentration than the first pass to the liver. This recirculation would not occur if they had not been conjugated. Metabolism affects the excretion because without it, ethinyl estradiol would remain a hydrophobic molecule inside cells. The various conjugations make the estrogens water-soluble so they can be excreted, or in the case of methylation, which caps the metabolites, they will become less hydrophilic and will bind to estrogen receptors in numerous tissues, possibly preventing damage. This is a detailed look at the metabolic stages during Phase 1 and Phase 2 of the natural estradiol and estrone, along with the two options for excretion after the metabolism, either excretion through conjugation or absorption into the tissues where it binds to estrogen receptors and triggers gene expressions. It also demonstrates the possible formation of quinones, which are a potential for carcinogenic effects. Although the schematic is not exactly ethinyl estradiol, it gives a lot of insight into the pathways and is quite useful. The excretion of the estrogen metabolites is metabolism dependent due to the hydrophobicity of the molecule. After undergoing conjugation reactions in Phase 2 of the metabolism, the conjugated glucuronide and sulfate estrogens are more hydrophilic or water-soluble and are easily excreted through the bile ducts, leaving the body in the forms of urine and feces. In the case of ethinyl estradiol following oral administration, there is a considerable amount of excreted metabolite in the feces, up to 62%, compared to the urine, which is 38%. There is a small amount, 6%, of ethinyl estradiol that is detected in the urine that is untransformed, as well as 9% of orally administered ethinyl estradiol appears as untransformed in the feces. However, in the urine, the majority is conjugated to the form of glucuronides, 80%, compared to the small portion of conjugated sulfates. The parent toxin is ethinyl estradiol, as that is a toxin that is originally absorbed into the body through oral intake. However, it is unknown if this is the only ultimate toxicant or if the metabolites are also ultimate toxicants. One possible mechanism is the investigation of 4-hydroxyl ethinyl estradiol as the ultimate toxicant. Through oxidation catalyzed by cytochrome P450 3A4, an additional hydroxyl group can be placed at the carbon 2 or 4 position, introducing another nucleophilic site. While 2-hydroxyl ethinyl estradiol is the most quantitatively dominant, 4-hydroxyl ethyl estradiol is so far the most carcinogenic and genotoxin metabolite. In this scenario, the toxin, ethinyl estradiol, is targeting the hepatocytes in the liver, which are epithelial tissue cells that make up 60 to 80% of the liver's mass. The cells can grow in long chains and form layers on top of themselves, which can become quite useful for conducting some of their functional roles. The hepatocytes are very multi-talented cells and are physiologically involved in metabolism, synthesis, and detoxification. Hepatocytes are responsible for the metabolism of lipids and carbohydrates, as well as the detoxification of drugs and insecticides, such as the reaction of ammonia into urea. They are responsible for the synthesis of proteins such as serum albumin, lipoproteins such as HDL and LDL, lipids for export, as well as producing and secreting bile. However, it is important to keep in mind that with respect to the toxin, the target is more specifically the estrogen receptors either on the exterior of the hepatocytes as well as possibly on the nucleus. There are two main estrogen receptors, alpha and beta. In the hepatocytes, the estrogen receptor alpha is dominant and once activated can assist in the numerous synthesis and transcription functions of the hepatocytes. In ideal conditions, the estrogen metabolite would bind to the estrogen receptor, activating it, and in turn activating the transcription of a particular gene. However, this is not always the case. As with other receptors, non-covalent interactions are often observed in which the metabolized toxin will temporarily bind to the estrogen receptor, causing a target dysfunction, such as preventing the receptor to activate and transcribe DNA. If a variation of estrogen binds to the estrogen receptor through hydrogen bonding, for example, then the receptor will not be activated. In the hepatocytes, this could result in an alteration in the production of the high-density lipoprotein, potentially causing problems with cholesterol, as it will not be dispersed as quickly if HDL is not present. Estrogen, binding either genomically or non-genomically, will induce transcription of genes such as MAPK and CCNDS, which result in cell proliferation, and if left unregulated, could result in the replication of perhaps many mutated cells. A second possible route of toxicity requires another metabolism. Once again, note that these diagrams are for the estradiol, however, the ethinyl estradiol functions in a similar fashion. While 2-hydroxyl ethinyl estradiol is the most quantitatively abundant metabolite hydroxylated, it is a weak carcinogen. The 4-hydroxyl ethinyl estradiol is the most genotoxic of the metabolites, regardless of the small quantity that it is produced. If this metabolite is exposed to a second oxidation instead of conjugation by methylation or glucuronidation, 3,4-quinones are formed as shown by the diagram to the bottom right. These quinones produce a site of nucleophilic attack by DNA as well as the radical species superoxide as a byproduct through reduction and therefore can cause numerous problems, such as lipid peroxidation, DNA adducts, damage to DNA and proteins, causing target destruction of the hepatocytes. The covalent interaction between the ethinylestradiol quinone and DNA results in the formation of an abasic site that will need to be repaired with the correct base before replication and division of the mutated gene occurs. If the ethinylestradiol metabolites undergo the second oxidation before they are conjugated, glutathione S-transferase, being a nucleophile, will bind to the quinone and cap the oxygen groups, allowing for the excretion of the toxin in order to avoid destruction of the DNA transcription. Ethinylestradiol metabolites are hepatocarcinogenesis promoters. Therefore, there is the risk of increasing the development of liver tumors in women. This is harmful at the organismal level as well as for the entire function of the body. If the liver is damaged, the metabolism of any molecule becomes jeopardized, bile is not produced, and toxins will not be broken down. Ethinylestradiol is also known to induce and increase the sex hormone binding globulin levels, which is a protein synthesized in the liver. As you age, your sex hormone binding levels tend to rise as your production of sex hormones such as estrogen decline. This results in the continuous production of SHBG, which binds to what little natural estrogen is remaining and reduces the bioavailability to the cell. In general, unless the synthetic ethinylestradiol is given in a high dosage numerous times or the metabolite is of the toxic variation, the metabolism of the estrogen metabolites uses the toxin for therapeutic effects, such as inducing the synthesis of HDL, which transports cholesterol away from your heart. Excess estrogen can cause birth defects, abnormal sexual development, problems with the nervous and immune system, and as discussed through DNA mutations, causing ultimately cancer. Ideally, if the metabolites are conjugated properly, the estrogen binds to the estrogen receptors and produces the expected transcription genes for female characteristics.